Have you ever wondered how proteins are produced in our body? This intriguing process, known as protein synthesis, happens constantly in our cells to make proteins that are essential for various cellular activities. From enzymes required for metabolic reactions to hormones that control body functions, proteins play a critical role in our biology. So how does this process happen? It boils down to two primary steps: transcription and translation. Transcription occurs in the cell nucleus and involves three phases: initiation, elongation, and termination. During initiation, enzymes come into play. Topoisomerase unwinds a portion of our DNA, which is usually tightly coiled. Following this, DNA helicase breaks the hydrogen bonds between the two DNA strands, freeing them up. DNA polymerase then binds to a promoter sequence on the leading DNA strand. Elongation is next. Here, RNA polymerase begins to produce a messenger RNA, or mRNA, using the DNA strand as a template. This process involves matching corresponding nucleotides, guanine with cytosine, adenine with uracil, and vice versa. This continues until the entire DNA segment of interest has been read. Then comes termination. Once the DNA segment is fully read, the newly formed mRNA strand peels off. It's now called pre-mRNA and undergoes RNA splicing, where introns or non-coding sequences are removed. Only the exons or coding sequences are kept. The resulting mRNA then leaves the nucleus, heading to the cytoplasm, where the second main step, translation, takes place. Translation involves reading the mRNA to create a specific protein based on the genetic code. Like transcription, it also involves initiation, elongation, and termination. During initiation, the mRNA binds to the ribosome at the start codon, which is always OG. A transfer RNA, or tRNA, carrying an amino acid and an anticodon complementary to the start codon binds to the mRNA. The first amino acid is always methionine. Then, during elongation, multiple tRNAs carrying corresponding anticodons bind to the mRNA, forming a polypeptide chain. Finally, termination occurs when a stop codon is reached. Translation halts, and the newly formed polypeptide is folded into a protein. So, there you have it. The riveting process of protein synthesis that happens within our cells every single day. From the unwinding of DNA during transcription, the creation of mRNA, to the formation of a protein through translation, it's a process that exemplifies the incredible complexity and efficiency of our bodies at a microscopic